Hello.
Yes, sir.
Jutika ma'am, can you hear me?
Yes, audible, sir.
Ma'am, I have sent the presentation. Kindly please check.
Yes, sir. I will do it, sir.
Okay. Thank you, ma'am. Thank you.
A very good afternoon to all. So today we are again going to start the second session. Hope you have gathered a lot of information in the first half talk. Now today we again have for the second session Dr. Ranjan Kalita. Welcome, sir. Welcome to this Atal FDB program. And before I request
Thank you, sir.
Dr. Kalita, I'd like to introduce him before you. Dr. Ranjan Kalita is an assistant professor and head of the Department of Biotechnology, Assam Royal Global University. Dr. Kalita did his PhD from Tezpur University in molecular biology and biotechnology. His research area includes drug discovery from natural products, cellulose and its derivatives in preparation of biocomposites, novel drug delivery methods, food nutrition analysis, etc. Dr. Kalita had carried out a very significant investigation, a very prominent investigation on development of water purifying filter based on cellulose and its derivatives. One of his one of his research that was highly cited and got a lot of citation in different journals. Dr. Kalita has many publications in peer-reviewed journals, international journals, and he has delivered many invited talks in different seminars for different organizations as well as in different webinars. We are very much thankful to Dr. Kalita that he has given his consent to deliver this talk today. And his talk will be on novel nanomaterials for targeted drug delivery, the path ahead. So with this brief introduction, I would like to request Dr. R. Kalita for his presentation. Now over to Dr. Kalita, please.
Yeah, thank you, Dr. Deka, for the beautiful introduction that you have given. And I thank the organizers for inviting me to deliver a talk on the Atal FDP online classes, and keeping in reference to the topic from CIPET, I kept my title in line with the already structured, the topic, that is the novel nanomaterials for targeted drug delivery and the path ahead. So I think, I welcome all the participants out here today, and I hope we can go for a, go for the lecture, and we can interact briefly after that. So it will, be for the benefit of both of us, all the way. So thank you once again. And I shall, switch off my video because of a low bandwidth problem out here in this part, where the internet problem, network is very bad. So my audio will be on. So my presentation is there. So thank you. Okay, so we shall start on with the talk. So my talk is based upon nanomaterials. As we all know, nanomaterials are the talk of the day, and nanomaterial research have been one of the most widely yeah, hello. Okay, is one of the most highly carried out research in various parts of the world, and this hello. Okay, and these nanomaterial related research have garnered thousands of publications, and in various various research specific domains, be it for drug delivery, water purification, pharmaceuticals, agriculture. So wherever we go today, we find nanomaterials being processed and being synthesized every day for the betterment of humanity. So ma'am, may I go for the next slide, please? Yeah, thank you. So a brief introduction for all the joinees in this faculty development program. And then you have given my presentation to the organizers, and if you still need it, you can take this particular presentation from them. So like nanotechnology, we all know, is a pioneering field, and in this we actually develop materials which are in the size of 10 to the power minus 9, that is in the nano range, where the particles are within the ambit of 5 to 200 nanometers. So for calling these particles as nanomaterials, it is not just being within less than 1,000 nanometer, but those which actually falls in the range of 5 to 200 nanometer, we actually take them for the research purpose and for drug delivery, keeping it with that particular topic. Now, why should we go for, or why have we come over for nanomaterial research in the area of drug delivery? So you see, humanity or human beings have been confined to the earth only, and in this system there has been a lot of diseases that we have to overcome. If you look through history, you'll find a lot of diseases which have haunted us, be it plague, tuberculosis, and in the recent cases you will find corona invading all our lives, and almost all of us have been infected once or twice. Then cancer is one of the most widely fought after disease, and it is believed that 1 out of every 10 individuals will be having cancer disease in their life. So it is a very complex situation in which humanity exists, and therefore, in such a situation, in such a pedestal, it is important for us that we devise certain ways in which we can make our lives simple and disease free. So, nanomaterial research, and for that matter, the broad term that is nanotechnology, comes into play. Now, again, you see, different diseases needs different ways of handling, and therefore there has to be different routes for administration of the drug. Now when you are speaking about routes for delivery, we shall anyway be going into that details later on, but then generally at home when we have diabetes, people they take drugs, they take tablets, and people have gastritis, they take tablets, pen 40, for diabetes, glycomed. So there are different kinds of drugs for pressure, I mean hypertension. We go for taking different kinds of drugs, slow, then Almin. So this kind of drugs we are taking it. But then if you look into these drugs, so these are in the form of tablets which are much bigger. We can see them, we can hold them, and we can easily feel them gulping, I mean going down our, I mean esophagus. Now the thing is that all these drugs, do they work fine for, and their activity stays there for 1 hour, 2 hours, 6 hours, 8 hours, 12 hours, or even for a day. However, for certain diseases, this form of uptake is not advisable because they do not lead to the reduction of the disease or for curing of the disease. So for them, we have to go through other routes. And therefore, finding out novel routes for drug delivery is one of the main essence in nanotechnology research. And also, for that matter, for the system to take up the drug is of utmost importance. So therefore, a true drug delivery system is with a formulation, or it can also be a device which allows the introduction of active ingredients into the body to improve their efficacy, safety, and the I mean, and we have to control the particular drug, and I mean we can control the particular drug amount. Then again, we can control the time and release of the drug in the particular site of action, and of crossing the various biological barriers so that these drugs of interest can move into the particular target. So therefore, a drug delivery system must encompass all these particular characteristics to move the drug into the target. Now, recent advances in nanotechnology have made people do research in nanoparticles, and these nanoparticles, they have the potential to improve the stability and the solubility of the encapsulated substances, and they also promotes their transport across the membranes. And not only that, nanoparticles also help in increasing the circulation time so that the drugs are effective. Then comes a question like, oh, why should you go for nanoparticle research? Now, if you take a piece of brick, you'll find that length, breadth, and height wise, it has three areas, and therefore there is a surface area which is there. But if you break it up into millions of particles, so each of the particle will be having a surface area. So that is the most important advantages in nanoparticles. They represent a diverse and a huge surface area for these drugs to be loaded and to be put in effectively into the system so that they can target the particular site. Now, nanomaterials hold promises to improve the disease diagnosis and also in the treatment of specific disease. So they are very specific, and we can actually direct the nanomaterials for targeted drug delivery and for a particular disease And we'll see, I mean, the applications of nanomaterials in the later part of this talk, wherein they have been applied not only for, I mean, I have only taken in a few points, but they have been applied in a huge diverse areas. So the polymeric nanoparticles can be synthesized, I mean, from natural sources or synthetic materials, and these substances must be biodegradable as well as biocompatible, and only then they shall be feasible in us, because if they are not biodegradable, then they will pose an environment problem, or even when they are inside us, if they do not degrade, they will stick to us, and later on in life they have the probability of causing diseases in us. Now they should be biocompatible, because if they are not biocompatible, again our body will not accept them, and there shall be various response against these nanoparticles subsequently that will actually pose a problem in us. Madam, next slide please. Yeah, thank you. So polymeric nanoparticles again can be formulated to enable the precise control of various features, and they are actually very good delivery vehicles, as they have been found to be biocompatible and also have simple formulation parameters. Okay, so we can go for easy formulation of them, of these polymeric nanoparticles, and we can go in for designing of them, and because they actually can take incorporate a lot of these features, and besides these we'll again speak about this functionalization of the nanoparticles, like how actually different things are added into the nanoparticles to make them more specific and targeting the particular site. Now, polymeric nanoparticles have a variety, I mean, have variable drug delivery capability. So these polymeric nanoparticles, which we are developing, they can actually go in for developing, delivering of different kinds of drugs at different target sites. Now, they are also structurally stable, as they have various porous characteristics. So they are basically porous, and they can be engineered for the required size and porosity. So this is where we can actually excel, because if we know that a particular nanoparticle can be broken up and it can be incorporated based upon its size, we can design and synthesize them as such, so that they can take the drug of our choice and cross that particular biological membrane and enter into the target site. Because whenever we put anything foreign into our system, our immune system will always come into play, and they will also have a role. So that will of course come later. Now, various functional groups are present on the surface of the polymeric nanoparticles, which can be modified with different targeting ligands. So, as I had said, we can actually functionalize the nanoparticles by using different functional groups, as well as other substances which will make them more effective and specific. So the polymeric nanoparticles are synthesized using various techniques, and nanoprecipitation, emulsification, gelation, microfluidics, which results in different final products. So again, we'll be having speaking about them, the nanorods, nanospheres, all these things. Ma'am, the next slide, please. Yeah, thank you. The inorganic nanoparticles are precisely formulated, and they can be engineered to have a wide variety of size, structure, and geometries. So these things are very important, because ultimately they will give us the product, as I had said previously, just like, and they are used in the form of nanospheres, nanorods, nano cages, nanocells. So these are all different ways in which the nanomaterials are synthesized, and the final products, they seem to be like cells or cages and spheres and rods. So based upon their structural observation, we actually name them as such, and since they're in the nano range, so we coined the term nano in front of the particular word. Now, the stability of a nanomaterial is influenced by the constituent material and its interaction with the environment. So this is very important, because when we have put a nanomaterial in our system, we want them to be stable, because If stability is not present, then it will be very difficult for us to take them or go out for, putting them as a drug delivery vehicle, because if they are not stable, they will not incorporate the drug into the particular target site. So that will actually again be more of uh, disadvantage to us than being of advantage. Ma'am, next slide. Yeah, thank you. So these are certain areas in which we can actually go for these biodegradable polymeric nanoparticles. So you can see, as I had said, drug delivery is one of the most vital areas in which we can propose the research or carry out research using these polymeric nanoparticles. Then we have the protective, for as a protective layer, we'll be putting out these nanoparticles because they will have a protective layer, as they need that to be stable within the system. Then they are also used in the preparation of biocatalysis, biosensors, which are, you can see almost in every domain, sensors coming up. And even the recent cases in corona times, you can see that in a country like China, where we had seen in a video that they have developed certain classes which are based upon sensors which can detect if a person is having corona or not from a distance. So you need not just go in for swabbing that person, or even going for, like taking samples, saliva samples, etc. for RT-PCR analysis. But actually, you can feel the person using that sensor from a distance, whether he or she is having a specific temperature. And then after he or she is having the specific temperature, you actually ask that person to, go in for further checkup. The next is development of biomarkers. So biomarkers are very important in the particular different cases. Different diseases like cancers, wherein different cancers, because cancer is a disease which actually can emerge in more than 200 cells of the human body. So therefore, in each of these cases, there will be certain molecules which will come up as when the cells are being when the cancer cells are being formed. So in this case, when they are being these markers are being generated, at that moment, if we take the blood of the sample, of the blood sample of the person, or of the infected person, then we can determine what is the particular stages and the particular person is having, or he is suffering from, and then we can actually go in for further treatment. Then magnetism or tracking and therapeutic agent, like different types of biodegradable polymeric nanoparticles which are based upon metals, and particularly they have magnetic properties, and based upon that we can actually devise and develop different types of therapeutic agents. Then biocompatibility is always there, because without being biocompatible, we cannot go for treatment of the specific disease using the nanoparticles, as different kinds of organs are there in the human system, and if any of these organ systems are affected, then it will pose serious problems. Then nanoparticles are also used in optical signaling, that is in imaging, and also in the detection of antigens, which is a very important aspect, because antigens are something which are foreign in us, and if these nanoparticles can detect the antigen based upon the activity of the antibodies actually, so that would have been it would be a very marvelous fact. But then research is going on in these fields, so again it will take time for all these technologies to come up. However, as of now, they are really been a boon to us actually. Madam, next slide, please. Hello, madam. Next slide, please. Yeah, thank you. So here in this particular slide, you can actually see out the different subclasses of nanoparticles with their various advantages and disadvantages, which have been referred at the bottom. You can see that in case of polymeric nanoparticles, we can have polymerosomes, dendrimers, then micelles, nanospheres, etc. So what are the advantages of them? So the first advantage is that these polymeric nanoparticles, they have precise control of the particles. I mean, we can have precise control on the particle characteristics. Then we can also have a flexibility in putting the payload. Payload means the contents which we can put in, that is the drugs and everything. Then easy surface modification. So there is like in the surface of nanoparticles, we can modify them, and based upon that, actually we can design our nanoparticles and we can functionalize them, and then we can put them inside the system. Then the next is that one of the disadvantages is that there is a possibility of toxicity as well as aggregation of these nanoparticles. So if there is an aggregation inside our system, if they aggregate, they will actually block the entire pathway, and that will actually pose a problem. So, and if they aggregate, then also there will be less, like these substances will be released, and that will actually pose a problem in drug delivery. Then toxicity. So one of the main problem in nanoparticle research is actually the toxicity, and various nanoparticles, due to their this surface surge and surface modifications, they turn out to be uh, toxic substances, and that actually pose a problem later on. Then there are these inorganic nanomaterials like silica, nanomaterial, iron oxide, gold, quantum dot. Although these nanoparticles have been found to be quite advantageous, yet they have certain, they have also toxicity problems. And one of the most important thing is solubility problem. So they have solubility limitations in solubility. Yet these inorganic nanoparticles have been found to be used in electrical, magnetic, and optical applications, and they have a very important characteristic in like the size and the structure, and actually we can get them in different sizes, structures, and geometry, so that is advantages for us. And lastly, they are used for theranostic applications. So theranostics means and therapeutic diagnostics. So there's a term, a new nomenclature which has been termed from these two words, therapeutics and diagnostics. So based upon that, based upon all these advantages, we can go in for application of the inorganic nanoparticles. However, the problem is the toxicity and solubility. So research must focus on these two particular aspects to see how they can go in without causing any problem to the system in which they are being injected. The next is the lipid-based nanoparticles. So the lipid-based nanoparticles are the liposomes and the lipid nanoparticles, which are created using different kinds of substances, cholesterols, and then other lipids, then emulsification process. So we are getting a structure in which the oil, that is a hydrophobic substance which is inside, and the hydrophilic area is outside. So these particular lipid-based nanoparticles have a lot of advantages. We'll go into that in the next slide. So here, these lipid-based nanoparticles. So an important characteristic is that the formulation is very simple, and you can use it anywhere. If you have a rotary evaporator in your laboratory, you can easily manufacture liposomes, and then they have a high bioavailability and also payload flexibility. So you can also determine the size, and based upon that, you can actually go for the payload, that is the cargo content of the particular nanoparticles. However, one major disadvantage of these lipid-based nanoparticle is that they have low encapsulation efficiency. So these drugs that are being encapsulated, so they can be, they are basically quite low than being thought of, and therefore you need to prepare a lot of them, or you have to go in for putting a lot of these nanoparticles into the system to function as they should be. Ma'am, please, the next slide. Yeah, thank you. So in the next slide, herein we are going for the various types of nanostructures for drug delivery. So we have been saying in the first two slides like different types of nano particles, nanospheres, nanorods, nanocases. So actually, and the sources from which they are prepared. So for here, I mean the types. The first, I mean we'll go in for only certain types, and I have not incorporated all the types of nanoparticles which have been synthesized till date. So the first one is the nanostructures from liposomes. So liposomes are small artificial phospholipid vesicles, and these are derived from phospholipids, various kinds of phospholipids such as phosphatidylglycerol, phosphatidylserine, phosphatidylcholine, etc. And these liposomes were first and have been used for delivery of various drugs. And based upon their lipid bilayers, the liposomes are divided into different types. So lipid, if you look into the, if you have seen the structures of the lipid-mediated nanoparticles in the earlier slide, you can see that they are actually different layers. Okay, so these liposomes are again divided based upon the layers into two types. So one is unilamellar vesicles. So these unilamellar vesicles, they consist of an aqueous core. So the inside part will be aqueous, and they will be surrounded by a lipid bilayer, and the inner core is separated from the outer core. Then comes the multilamellar vesicles. So these multilamellar vesicles, they are composed of several layers of lipid bilayers, and based upon this, they have been actually given the terminologies. That is unilamellar, one layer of lipid; multilamellar, multiple layers of lipids. Now, the liposomes are biocompatible, so that is a very big advantage for us. Then they are biodegradable, and they can compartmentalize and solubilize both hydrophobic and hydrophilic materials. So these liposomes are again rapidly taken up by the reticuloendothelial system in the body, which further takes them down for their degradation. Next, polymer-based nanostructures. The next slide. Yeah, thank you. The polymer-based drug nanostructures, we had already studied about a brief introduction of them in the second slide, the polymer nanostructures. So these nanostructures can increase the solubility of hydrophobic drugs. So that is one very big advantage. That is, they can increase the solubility of the hydrophobic drugs, and the polymers which are used must be, but yeah, this is an important point out here. That is, the polymers which are being used, they should be non-toxic and biocompatible. Now, in these cases, the natural polymers are basically chitosan, dextran, heparin, hyaluronan. So these are the natural polymers which are used, and the synthetic polymers are polyesters, polycarbonates, polyamides, polyphosphazines, etc. So these both natural and synthetic polymers are used in the development of the polymer-based nanostructures. Then nanocapsules. So these are again a different type of nanomaterials which are given the name as capsules because of their final end product. So these nanocapsules are particularly developed by emulsion process, and this emulsion process finally allows the formation of certain external polymeric membranes, and the drugs are particularly confined in the cavity of the nanocapsules. So these drugs can be loaded inside. So it is just like a particular capsule drug in which if you take the, for example, the bicosol drug, so you'll find that if you open the two layers, that is, if you twist it, you can open it, then you'll find that the drugs are being loaded in the inside, that is the inside cavity, which is a hollow part, and therefore they are surrounded by the external polymeric membrane. So you can take the core of that bicosal tablet as a polymeric membrane. Okay, so these nanocapsules, they help in increasing the bioavailability of various drugs, including proteins, peptides, and insulin. So this is because they can actually bypass several of the barriers, and they can move about to places where you want our drugs to be targeted and delivered, and therefore they increase the bioavailability and also helps in stabilizing the drug and distribution of the drug in the body. Ma'am, the next slide. Okay, thank you. So polymeric dendrimers. So dendrimers are certain structures which have been widely used in the designing of various nanoparticles, and also in in the delivery of different kinds of drugs. The dendrimers were first discovered by Tomalia. I mean, he actually prepared by these dendrimers, and he also coined the term dendrimer. So Tomalia did these experiments way back in the 1990s, early 1990s, wherein he suggested that dendrimers would be the next in revolution for drug delivery. But at that time, little did people realize that it was a nano form thing, and which actually have an immense role to play later on. So these dendrimers are nano-sized, and they are symmetric molecules having well-defined homogeneous and monodispersed structure, and they have tree-like arms or branches. So if you look into the structure of a dendrimer, of course, I didn't put the structure of a dendrimer out here. I forgot to put that. But if you look into that, you'll find that they have various branches protruding from the structure. So these branches, they can actually hold in a large number of elements, and they can go in for delivering of the particular agent which we need. So therefore, dendrimers are very important for drug delivery. So they have advantages like they are being nano size, then having a narrow polydispersity index, that is, if you look into their size and length, then they are stable molecular structures, and the availability of multiple functional groups. So when you have a large number of branches, you can also put in a large number of functional groups. So they are used in a wide range of applications, which are used in the delivery of vaccines, then enzymes, then genes, oligonucleotides, etc. Then ceramic nanostructures. So ceramic nanostructures are porous nanoparticles, and they are developed from biocompatible inorganic compounds such as silica, calcium, phosphate, alumina, etc. So these nanostructures are again believed to be of excellent carrier for drugs, various genes, and proteins. So these ceramic nanostructures are being usually prepared with those materials which are biocompatible. So silica, calcium phosphate, and alumina are there. But again, they have certain advantages and disadvantages, which we shall discuss in the later part of this particular presentation. Madam, the next slide, please. Metallic nanostructures. So metallic nanostructures are one of the most expensive, I would say, as well as sought after, because they are, I mean, they were newly discovered, and of course, it is more than, I mean, it is approximately a decade for this discovery, wherein the different kinds of metals like gold, silver, gadolinium, iron oxide, etc., are being used, and these metallic nanostructures are used because they have favorable optical and chemical properties. They can also be modified in using various functional groups. So that is why they are very important. And they are used for the delivery of genes, proteins, and peptides. So you can see the versatility that is, the thing that where they are being applied. And gold nanoparticles, they also help in the enhancing the stability, and they serve as scaffolds in drug delivery. So gold nanoparticles are one of the most, uh, you know, hot topics of research, wherein people have been using them for a quite a long time, and they have been able to make inroads into the treatment of different diseases. But then again, certain limitations are there, which we shall be listening to. Then the iron oxide nanoparticles makes up a majority of the FDA approved nanomedicines. So FDA is the drug regulatory authority of the United States of America, wherein every thing which is prepared for human uses, particularly in the medicine sector, has to be approved by the drug regulatory authority, and they will finally say whether your drug is applicable for the market or not. So that is very important. And interestingly, the iron oxide nanoparticles actually makes up more than the majority of the nanomedicines which have been approved by the FDA. Then we have the peptide-based nanostructures. So peptides, from the name, you must have realized that these are small segments of proteins actually. So if you take in the structure of a protein, which are made up of amino acids again, so these amino acids in between two of the amino acids, so if, for example, you are taking serine and proline, in between these two amino acids, they are being joined by a bond called the peptide bond. Oh, yeah, the peptide bond, that is a CONH bond. Now, when there are more than one peptide, say up to 8 or 10 of them, then these are smaller structures in linear in length, which we take them as a peptide based structure. So if they are more, that is, they become polypeptide, that is, two thousands of peptides are there or hundreds of peptides are there which have been joined together. So we coin them as a particular protein. But the thing is that these peptide based nanostructures, they have a very important role in drug delivery, and because they are biological in origin and they have high biocompatibility and degradability. So oligopeptides can be prepared by using various synthesis process at low cost. The nucleic acid based nanostructures. So nucleic acid nanostructures, which are based on DNA and RNA, are widely used in various drug delivery applications. So these DNA based nanostructures can be actually functionalized and have low immunogenicity. So immunogenicity is the problem when you will be having immune response against like a variety of antigens, and therefore we must make those kind of substances. I mean those kind of substances which usually invoke low immunogenicity. So you do not want to risk them. Okay, so therefore these kind of nucleic acid nanostructures can be used as they do not demonstrate again any high immunogenicity in the human system when they are being used. So, ma'am, the next slide, please. Thank you. So herein we have the last of the structures, that is the nanostructures, that is carbon-based nanostructures. So these carbon-based nanostructures, basically, you have heard about graphene, fullerene, and then carbon nanotubes. They are developed for drug delivery. So graphene is a multifaceted material which actually can be wrapped into spherical structures or even formed into one-dimensional or three-dimensional structures. Then the carbon nanotubes, due to the similarity with graphene, has also been widely used in the targeted delivery of drugs, genes, and proteins. So you must all have realized how important graphene is, and you all probably please try to remember, like in the year around the early part of this past decade, this graphene was discovered by a very young man who was doing his PhD along with his guide, and they actually got the Nobel Prize for this discovery. So graphene is very important for all of us, and they are also used in the preparation of nanostructures which can be used for drug delivery. Ma'am, next slide. Yeah, thank you. So in this slide, you can actually see the different methods for the preparation of the polymeric nanoparticles, and you can see that there are methods like solvent evaporation, emulsification, salting out, dialysis, polymerization, supercritical fluid technology, nanoprecipitation. And under each of these broad categories, the smaller boxes again denotes how they are actually made. Just like, for example, in solvent evaporation, in the first method, the emulsion is converted into a non-nanoparticle. And then the single emulsions, that is oil in water or water and oil methods, are being used for the uh, process of obtaining of the nanoparticles using solvent evaporation technique. So again, in case of polymerization, you can see that there are again different steps like microemulsion, mini emulsion, emulsion, interfacial, and controlled. Then, in case of supercritical fluid technology, you can see that they are actually very environment friendly and they have a rapid expansion of the this particular technology has been rapidly expanded because of their environment friendly nature and their use, and because of the instrumentation availability, and also because they are not that expensive. So in salting out, it is actually a modification of the emulsification process, wherein the different kinds of salts are actually separated using diffusion process. So if you look into this slide, you can actually relate the processes by which different kinds of nanoparticles can be prepared, and you can actually relate them to the different types of research papers, and you can see out how they are prepared, and according to your need, you should be going for that particular structure. You should not be like, oh no, this is a so good nanoparticle, this is so costly nanoparticle, I should be taking it for my research. No, it might actually fail you. But you should actually look into the literature to see how the different kinds of nanoparticles, they pose serious concern or for that matter, like how they are prepared and how they can be used for and actual delivery of the drugs, or even like how they can be used in case of the betterment of the human civilization. So in the next slide, that is uptake of nanoparticles, we will see how the different nanoparticles are taken up by the system. So broadly saying, this uptake is actually divided into two types. One is the active uptake and the second is the passive uptake. So the cell membrane, when we are actually putting the nanoparticles into a body, they have to go through the cells, right, for certain type of delivery. But then here comes a twist. That is, these cells, they have to allow the nanoparticle to enter into themselves, right? So how will they enter if they don't allow? So they cannot enter, and subsequently the proper treatment process will be hampered, and these nanoparticles will actually not help in the curing of the disease. So therefore, the cell membrane, and why this phenomenon actually occurs. So this actually occurs because the cell membrane is selectively permeable, and therefore passive diffusion is limited to small uncharged molecules which can travel down the concentration gradient. So this is very important for us because of the selectively permeable nature of the cell membrane. Passive diffusion has to be taken into account. The nanoparticles rely on active transport to cross the cell membrane. Again, you have to go for active transport because for the proper transport across the cell membrane, the nanoparticles rely on the active transport mechanism. Now here the nanoparticles tend to utilize the endocytic pathway in which the plasma membrane is folded into vesicles to engulf the nanoparticles on the cell surface and then release them intracellularly. So endocytic pathway is something where the plasma, if you look into your particular membrane, and if you fold the membrane inwards like into the inside of the cell, so you can see that they are being folded inwards. So there is an invagination. Okay, that for particular terminology out there, that is called invagination, and it is very necessary so that the nanoparticles are taken inside the cell to be crossed across the cell membrane. Then the type of endocytosis which is taken by a nanoparticle undergoes, which is taken up by the nanoparticle, and can actually affect its fate within the cell. And this is determined by numerous factors, including the cell type, the nanoparticle size, and receptor interactions. So again, there are various things upon which all these factors depends upon. So herein you can see that this various factors includes cell types, the size, and the interaction with the different kinds of receptors. In non-specific cell membrane interaction, the smaller or larger nanoparticles will be taken up either by phagocytosis or pinocytosis. So you can see that the uptake of the nanoparticle is either through phagocytosis process, which is usually done by the immune system, or pinocytosis is usually done by different kinds of membrane to dissolve in the liquid substance. Then the negatively charged nanoparticles results in either caveolin mediated or clathrin mediated endocytosis. So what are caveolin mediated and clathrin mediated endocytosis? Now, when we are saying about caveolin mediated endocytosis, these are actually clathrin independent processes. That means in clathrin dependent processes, what actually happens is that those clathrin mediated endocytosis are also known as receptor mediated endocytosis. So when you say a receptor, that means something on the cell which is holding them, okay, holding anything. So that means you're receiving it, okay. So this receptor mediated endocytosis undergoes uh, certain changes in which there will be inward budding of the plasma membrane. So your plasma membrane will be moving towards inside of the cell. Again it will come out, again move inside, again it will come out, and like that. So this inward vagination, this process also known as inward vagination of the plasma membrane, results in the taking up of different kinds of these nanoparticles. So this is the clathrin mediated endocytic pathway, and the other is the caveolin mediated. So caveolin is a clathrin independent process which involves bulb shaped and 50 to 60 micron plasma membrane invaginations. So these invaginations occur out here also, and they are known as the uh, caveos, okay, CAVOs. And these are again driven by, this process is again driven by internal membrane proteins, which are known as caveolins, as well as the peripheral membrane proteins called the caveins. So these two proteins are involved out here, and therefore they are, I mean, this pathway is known as the caveolin mediated pathway. The next is, okay, yeah. So the type of endocytosis a nanoparticle undergoes actually affects the fate within the cell. And ma'am, the previous slide itself, I have not finished it. Yeah, no. Again, previous. Yeah, thank you. So these nanoparticles in the type of endocytosis, the nanoparticles they undergo, I mean, the type of endocytosis that the nanoparticle undergoes also affects the fate of that nanoparticle within the particular cell, and it is determined by various factors such as the cell type, the nanoparticle size, and the interaction with the particular receptors. So in non-specific cell membrane interactions, very smaller and larger nanoparticles will be taken up either by phagocytosis or by pinocytosis process. Okay, so this is again very important. And lastly, the negatively charged nanoparticles results in caveolin mediated or clathrin mediated endocytosis. So this I had already referred to when I was speaking about endocytosis a few minutes ago. Okay, ma'am, the next slide now. Thank you. The caveolin mediated endocytosis, they occur in molecules which are smaller than 60 nanometers. And out here, lipid raft are being used, which actually creates specialized vesicles after engulfment. So this form of endocytosis process is most common for nanorods, and the clathrin-mediated endocytosis is usually taken up for nanoparticle uptake and generally in non-specialized mammalian cells, where these are actually receptor-mediated endocytosis, and here hydrophobic or electrostatic interactions takes place between the nanoparticles and the cell membranes. So this form of interactions, the bonding between them, these are very essential. Lastly, the induction of this endocytic pathway is influenced by nanoparticle properties such as stiffness and size. So again, how stiff the nanoparticle is, what is its sizes, whether they are soft or they are hard. So these things also come into play. And for all of you, it is the stiff nanoparticles are most easily taken up, okay, than the soft nanoparticles. So therefore, that is very important out here. Yes, ma'am, the next slide. Yeah, thank you. So in this slide with the diagram, you can see the caveolin mediated endocytosis process wherein the plasma membrane actually dips inside. That is, you can see a bulb-like formation of the plasma membrane. So if you can look into the structure, so in the first case, wherein different rod-shaped and small round-shaped nanoparticles are coming out, they are moving inside that bulb-shaped structure. So these are again due to the different types of proteins, the caveola and the caveolins, which are there, and therefore this entire process is known as the caveolin-mediated endocytosis. So once these nanoparticles are formed, I mean they are move inside these invaginations, these bulb-shaped invaginations, they are then separated into a particular sphere, and they will move inside. Okay, so they this process, because of the role played by both the caveola and the caveolins, this process is known as the caveolin-mediated endocytosis process. In the next one, that is in the middle one, you can see that it is a clathrin-mediated endocytosis process. So this is a receptor-mediated endocytosis process. So here you can see a Y-shaped structure out here in the cell wall. So this Y-shaped structure, these are particularly receptors which are located on the cell wall, and these receptors, they actually take up the nanoparticles. They receive the nanoparticles. Okay, so once they receive the nanoparticles, they through the clathrin-coated pit will move inside, okay, and that vesicle will formation will take place. So within that vesicle formation, you can actually see the different types of nanoparticles which have been taken up by the receptors. They are located inside, and subsequently these vesicles are the structures which will be used for further analysis. So again, apart from this clavulin and the clathrin-mediated endocytosis, there are also independent endocytosis processes as well as pinocytosis process, which also helps in the taking up of the nanoparticles. So you can see the legend out here at the bottom, which says that upon interaction with the cell surface, the nanoparticles, depending upon their surface size, shape, and charge, will be taken up by various types of endocytosis or pinocytosis process. And these are all non-specific interactions. Therefore, these are the different processes by which the nanoparticles will be taken up. Then, in the next slide, the next slide, please. Yeah, thank you. So in the next slide, you can see that there is an ionizable nanoparticle, which is being again taken up. I mean, when the ionizable nanoparticle enters the cell, so they remain trapped within the vesicular compartments or the endosomes. which actually traps them inside. But this nanoparticle must undergo escape mechanism so that they can function. So once they move out into the cell, move inside the cell, they will be trapped in. But due to different mechanisms, they will be subsequently allowed again to escape. Okay, so this is a very important process in which this endosome will be taking up the nanoparticles, but then the nanoparticles also find a way to escape out from these particular cells. Ma'am, the next slide. Okay, so delivery and biodistribution of nanoparticles. The distribution of uh, the nanoparticles and their clearance involves various processes. So the biodistribution. Hello. Yeah. Okay.
Yes, sir. Audible, sir.
Yeah. Thank you. So in here, like in the delivery and the biodistribution of the nanoparticles, so you can see that the nanoparticles, the distribution and their clearance are very important. So in this particular like mechanisms, so there are various processes by which the nanoparticles are taken up, they are distributed, and they are cleared from the system. So we shall be going into that. So for the first and the most important step is the extravasation process. So extravasation is something when it is actually leakage of a fluidic structure. Say suppose there is a blood capillary and the capillary burst, and all the blood inside that capillary will be moving out along with all the contents. So that is extravasation. So it is the first step of a nanoparticle in circulation to reach the target tissue. So the nanoparticles, when they are moving inside this particular uh, ways or vehicles, or they will, what they will do is they will undergo the extravasation process, and then they will try to reach their target tissues. This extravasation, however, can be altered by different types of characteristics of the nanoparticles, including sizes. So small nanoparticles, they generally cross the capillary walls more easily than the bigger nanoparticles. So therefore, nanoparticles can be distributed across organs in a size dependent manner. So the more smaller the nanoparticles, the more easier it is for them to cross the capillary walls. Then the nanoparticles which are injected intravenously, so intravenous injection of nanoparticles, basically they are taken up and then they are accumulated in the liver and the spleen, and subsequently from here their biodistribution process will start. But here there is a problem. The problem is that once they come over to the liver and subsequent distribution will be there will be a loss of function. So that is what is not desired. Now, in the next case, when the nanoparticles are subcutaneously or intranodally injected, they accumulate in the lymph nodes. So intranodal injection is something like when you inject it into the lymph nodes. Okay, so this is a recent trend in which you can find them being used so as to inject the drugs directly into the lymph nodes. It is mostly used in cancer diseases, treatment of cancer diseases, metastatic cancers. The alternate, various other alternate administration routes are also present, which enables the nanoparticles to reach the lymphatic system prior to the systemic circulation. So if the nanoparticles can move to the lymphatic system before moving into the systemic circulation, like moving the spleen and the liver, they can be more beneficial in certain immunotherapeutic applications. Then nanoparticles can also be delivered using the pulmonary mode of travel. So herein they can be directly put into the system through the process of inhalation. So this inhalation process, it avoids the exposure to the systemic circulation prior to lung delivery. So therefore, it avoids the hepatic first pass metabolism and thereby increasing the delivery of the dendrimers based nanoparticles to the lung and lymph node as compared with intravenous delivery. So herein you can see that there's a terminology that I have used, that is a first pass, hepatic first pass metabolism. So just as here in the point number 2, I have said that nanoparticles which are injected intravenously, they accumulate first in the liver and the spleen. So therefore, out here also you can see that the nanoparticles which are injected through the pulmonary mode through inhalation, they will directly move into the they will directly move into the lungs without moving into the liver or the spleen. So this is actually not required for us if you want, if we want our nanoparticles to perform the best of the function. So therefore, this process is very important, and if we can design them in such a way that they can actually bypass the systemic mechanism, then they will actually be of help to us. Now again, this inhalation process, most of the dendrimer based nanoparticles are being used in this pulmonary mode of delivery, and these dendrimer based nanoparticles have been directly used to be delivered into the lungs and to the lymph nodes as compared with that of the intravenous delivery. So therefore, when we are speaking about these processes, so it seems that it is easy process. Then, ma'am, the next slide. So although we had said that it is very easy, it is very good if we go in for the process of improved delivery into the lung tissues through inhalation, yet there is also a problem. The problem is if you move through that route, that is the oral route, then through the inhalation mode, which is basically the pulmonary mode, then the mucus lining is present. Okay, as well as there are certain surfactant which are present, and all these actually pose as a physical barrier for delivery of the drugs to the lungs. So therefore, we have to design the nanoparticles in such a way that they can be taken up through the pulmonary mode, and they can actually evade the different kinds of responses or blockages like mucus and other pulmonary surfactants. Then clinically approved nanoparticle formulations can also be administered intramurally or intravenously, and this actually has been there are many such nanoparticles which have been developed, and they are already in the clinical phases, and a lot of studies have been going on to see how they actually can be moved in or can be taken in the system. Now, the nanoparticles can be modified by addition of various targeting moieties to the surfaces to direct their delivery. So, when we are saying that they can be modified by addition of targeting moieties, that means we can actually modify the nanoparticles. You can remember, if you remember, in before we had said that the nanoparticles have a large surface size, surface area, and based upon that, they have also the binding ability to different groups. So in this case, also the nanoparticles are taken up, and different kinds of moieties can be added to the surface of the nanoparticles so that they can be easily delivered. So these different kinds of moieties are the antibodies, glucoses, folates, etc. And such active targeting, that is targeting with these different moieties, it improves the distribution of the nanoparticles within a target tissue. So that is where it is very useful. Now herein again, I'd like to point out, like, these moieties previously I had said we had discussed about, or I had presented about how these nanoparticles can actually target the antigens. So it is like they can bind to these antibodies and then subsequently perform the action like antibodies, and they can target the different kinds of antigens. Another thing is nanoparticles are also useful in binding to antigens and acting as an antigen presenting cell by taking those antigens to the T cells. Okay, unlike the general mechanism wherein the macrophages will be playing, and as well as the B cells will be playing in the immune response. Now, the nanoparticles, if they have to reach the central nervous system, they must use receptor mediated endocytosis. So, this is a very big problem, that is the incorporation of the nanoparticles, and for them to be directed to the central nervous system. So there has to be certain receptor mediated endocytosis, that is they have to be taken up by the cells, endothelial cells of the blood brain barrier, and once they are taken up by the endocytosis cells on one side, they will be exocytosed to the other side. That means they are taken inside a membrane and they will be put in to the other side of the membrane. So receptor mediated transcytosis. So transcytosis means when you are moving something through a particular membrane, from one side to the other side by taking them up. So this transcytosis process is an effective way to deliver different kinds of therapeutics to the brain, and also it helps in the infiltration of tumor tissues. Although the activity of these nanoparticles in a tumor is quite debatable, because moving inside a tumor and then performing the activity over there, and it involves a lot of Herculean task, because the microenvironment within the tumor also has different kinds of barriers which actually prevent the nanoparticles from performing their function. Then the oral delivery is the most highly preferred means of drug administration. We all know that, because anything we all always refer to being taken up by the through the mouth, rather than taking an injection and moving into the pharmacy, going to the pharmacy or to the hospital. Because many a times if you see oral delivery, if we take a drug through oral delivery, we can take it at a home, we can take it at our office, anywhere. Okay, we just need the tablets. But if you're thinking of taking an injection, you cannot do it by yourself. So you have to move to a, a pharmacy or to a hospital, so that a trained person can effectively incorporate the drug into your system. So that is very important again. So therefore, this oral delivery of the drug, which is most highly preferred means of drug administration, is often a failure, because different barriers are posed by the gastrointestinal tract. Okay, so as such, it is quite a difficult route for the nanoparticles, and there are a lot of challenges which are posed by the gastrointestinal tract, and therefore these all present barriers in achieving the particular biodistribution via the oral delivery. So therefore, even though we say that oral delivery is very important, or it can be used, or it is preferred means, yet in this case, due to the problems by the GI tract, it is not that feasible. Madam, next slide. So in this figure, excuse me. So in this figure, you can see I've shown the figures for extravasation, then clearance, and then in the third one at the bottom left, you will be seeing about the tumors and about a mucus layer. So in the middle row, if you look into in the first middle column, so in the first row is the different sizes of the nanoparticles, that is, which are larger than larger than 150 nanometer, in between 20 to 150 nanometer, and less than 5 nanometer. So again, you have different shapes of the nanoparticles. Some of them are round, spherical, some of them are rod shaped, then charged, positive charge, neutral charge, negative charge, then coated. So nanoparticles are coated like as if they are wearing a t-shirt or something like that on top of them. And lastly, you have the targeted nanoparticles, which would be having different types of functional groups around them. So if you look in the legend, you can see that the spherical and the larger, I have explained out here, the spherical and the larger nanoparticles, they marginate more easily during circulation. That means they can actually relate to the type where they are moving and how they can actually move out. Whereas the rod-shaped nanoparticles, they extravasate more readily. So that means the rod-shaped nanoparticles can actually try to move out from the circulation. Okay. However, these spherical and larger nanoparticles, they generally do not tend to move out. I mean, do not go for extravasation. Then, the uncoated or positively charged nanoparticles are usually cleared more quickly by the macrophages. Okay, so this is another important thing, that is the uncoated nanoparticles or the naked nanoparticles, and those nanoparticles which are positively charged, that is, they have a positive charge, they are cleared more quickly by the macrophages. So the macrophage, is the first line of defense in the system, and they are actually like the foot soldiers. So they will be seeing out what is coming up and attacking the system, of which is a foreign particle. So the moment they see a foreign particle, they will immediately go and bind upon them, and they will go in for the phagocytic activity, and subsequently they'll be presenting these particular cells. So therefore, if a nanoparticle uh, is uncoated and has a positive charge, then they'll be taken up by the macrophage and they'll be immediately cleared from circulation. Again, in terms of distribution, the rod-shaped, neutral, charged, and the targeted nanoparticles can penetrate tumor cells more readily. So you can see that the rod-shaped cells, the neutral charge cells, and the targeted nanoparticles, they can actually penetrate the tumors more readily, whereas the positive charge, smaller, and coated nanoparticles can easily traverse the mucosal barrier. So easily traverse the mucosal barriers means they can easily move in wherever, even though the mucosal barrier is present, and that is very important for us. Madam, the next slide. So in this slide, we'll be going through the different barriers in biodistribution of nanoparticles. So it is not like smooth journey for the nanoparticles in the process of delivery of drugs, but it is a very Herculean task for them too. And therefore, you'll find that there are different barriers actually in the body which prevents this smooth transaction. So out of these, the various physical and biological barriers, they have important role in preventing the nanoparticles from the effective biodistribution and delivery of the desired drug. So physical and biological barriers play an important role in prevention of the nanoparticles from functioning properly. So these barriers also limit the fraction of administered nanoparticles, which are used to reach the target therapeutic sites. Then again, there are different biological barriers which are encountered by nanoparticles, which depends on the route of administration and also the type of disease and its progression. So the biological barriers encountered by nanoparticles, they have different types of administration routes, and they also prevent in the type of treatment and of the disease and the progression. So then coming to the fourth point, that is during circulation, different factors are present, such as excretion, blood flow, presence of the phagocytic cells, which can also reduce the stability of the nanoparticles and their delivery. So these are other few important points, that is the excretion process, the blood flow, and the phagocytic cells, presence of the phagocytic cells. So that means if there is an immune response, then also the stability of the nanoparticles is at stake, and subsequently their delivery is a problem. Then the nanoparticles which are diameter less than 10 nanometer are observed to be rapidly eliminated by the kidneys, whereas the nanoparticles which are larger than 200 nanometer, they also has a risk in activating the complement system unless they are engineered. So that means unless modified using different groups on the surfaces of the nanoparticles, those nanoparticles which are quite smaller, they can actually be eliminated by the kidneys, and the larger ones, they can have the risk of activating the immune response, that is the complement system. Want themselves to be moved out, be operated, coated. So this pegylation process, it improves the circuit. These nanoparticles have been pegylated, that is, and the coating of PEG on top of them. So these nanoparticles are now no more recognized. Okay, so these actually will help them in being in circulation for a longer time, and they will have a higher solubility, which will also help these nanoparticles help the nanoparticles from being degraded by enzymes as well as by antibodies, which can induce degradation or secretion and clearance, subsequently clearance. So therefore, such kind of modifications of the nanoparticles is of very importance if we want to prevent their rapid excretion from the body. Now, the surface modifications can be done by just like we had discussed in the previous case, like in pegylation, or even by other cloaking techniques. So cloaking means you're putting a a, a coating on to the particular substance. So you're putting the substance where a trace. So that is called a cloak. So this cloaking technique allows the nanoparticles to avoid the recognition and clearance system. So once you actually mask them using a surface modification system, then you also prevent the nanoparticles from being recognized as well as from being cleared from the system, and this will actually allow the nanoparticles to be much more stable, as they won't be degraded or they are no more unstable. Now, next, in the next point, you can see that the lipid-based and polymer-based nanoparticles are the most susceptible to instability and aggregation, both in circulation and in storage. So these nanoparticles, which are lipid-based and which are polymer-based, they are most susceptible to instability. So they are quite unstable, and they also tend to aggregate. Okay, when in circulation as well as in storage. So if they aggregate during circulation, that is pose a problem, and even if they aggregate in storage, that will also be a problem, because while in storage, if they aggregate, we cannot keep them for a longer time. That is, the stability is no longer in question. I mean, no longer present. To improve the specificity, again, to improve the specificity of these nanoparticles, so we have to put in various excipients such as lipids, cholesterol, and other pegylated lipids, which with the help of which they are formulated. The nanoparticles can be formulated, and that will actually help them to increase the stability. Now, if we look into the bloodstream, the nanoparticles, they experience various flow rates. So if you look into the human physiology, you will find that the flow of blood into the capillaries, through the veins, and from the head to the toe, if you look into the blood flow rate, so you can see that it is quite different. They do not flow in as they move in through the veins or through the arteries. So therefore, this blood flow rate will also have an impact upon the nanoparticles. Okay, so therefore, these nanoparticles has to be controlled or has to be made manufactured in such a way so that they can uh, they can stay stable, or they can perform the activities even when different flow rates are observed. Now, in the next point, you can see that nanoparticles which are in circulation, sometimes they comes in contact with various cells and biomolecules suspended in blood. So the blood is not just a red liquid, it has different types of substances. If you look into the total the homeostasis process, you'll find that through this these homeostatic stem cells, when subsequently blood will be developed, you'll be finding that there are different kinds of cells which are found. Along with them, the immune cells will also be there, the RBCs would be there, the platelets, the WBCs, all these will be there. So therefore, uh, when the nanoparticles which are in circulation, they will come in contact with different kinds of cells and biomolecules which are suspended in the blood, and this non-specific adherence to the serum proteins as well as to the lipids may lead to the formation of corona on the nanoparticle surface. So these corona formations sometimes dictate the distribution of nanoparticles and compromise the stability of the nanoparticles as well as its contents. So therefore, to prevent the nanoparticles from degrading, or to prevent the improper distribution, the corona formation has to be eliminated. So that is something which we do not want. So herein, corona does not refer to the COVID-19 SARS-CoV-2 virus or that particular disease that it causes. So corona out here reflects the, whenever you are actually crowned, a king is crowned and he is put the crown on his head, so that is called the coronation process. So that is the corona. Okay, so out here, when the nanoparticles, they get in touch with different kinds of cells and biomolecules which are present in the blood, so they get designed using these substances, and once they are designed using these substances, so that looks like a corona in them. Now, the clearance of the nanoparticles from the circulation is influenced by different physiochemical properties, which results from interaction with the mononuclear phagocytes or the reticuloendothelial system. So again, the clearance of the nanoparticles from the circulation is influenced by different physicochemical properties, and this also results from the interaction with mononuclear phagocytes and the reticuloendothelial system. Okay, ma'am, the next slide. Thank you. So these systems are basically the monocytes, the phagocytes, the dendritic cells, which takes up the nanoparticles and they accumulates them in the spleen and the liver. So therefore, this immune response that takes place will ultimately take up the nanoparticles, will engulf them, will phagocytose them, and subsequently will take them to the spleen and the liver for further activities. Now, whenever there is any surface charge, in the nanoparticles, whether it is a positive charged nanoparticle, negatively charged nanoparticle, or neutral nanoparticle, then The cationic nanoparticles are actually the most rapidly cleared, okay, followed by the anionic nanoparticles. So the first one will be the cations, the second will be the anions, whereas the neutral and the negative, slightly negative nanoparticles have the longest half-life in circulation. So when you actually take up these nanoparticles for circulation, you have to look into whether the presence of the charges in them, and we have to see that the neutral nanoparticles and the slightly negative nanoparticles are in play because they have the longest half-life in circulation. Then, to minimize the clearance, the nanoparticles are designed with modification on the surface. So this is what I was referring to as the corona in the previous cases. So even though they mix with some unwanted substance and make them as the corona, and because of which they become unstable, out here we design them in such a way so as to prevent them from being actually taken by different other cells. Okay, so this is something which we manufacture. So this also helps in the reduction of the interaction of the nanoparticles with the phagocytic cells. So when we actually mask them, we also prevent them from being recognized. So in this way, they are, free to perform their function. Then the physical barriers to the nanoparticle distribution includes different tight junctions which are actually present in our system. If you look into the nervous system, and this among the endothelial and epithelial cells of the blood-brain barrier and the GI tract. So the physical barriers are basically the tight junctions which are present in the system. Now, once the nanoparticle reach the target site, they have to navigate the microenvironment, which presents various obstacles in the form of chemical and physical barriers. So again, the nanoparticles are not just free, they also have to navigate in that particular microenvironment where they are targeting the particular disease site, and these in that particular microenvironment, they also have different kinds of chemical as well as physical barriers. Now, after evading all of these chemical and physical barriers, the nanoparticles will be finally able to carry out their activities. Ma'am, the next slide. Thank you. The nanoparticle penetration and stability are mostly challenging in the solid tumors. So tumors are something which are actually cancerous, and they can be benign, they can be metastatic. But of course, benign cancers, benign tumors do not pose any problem, and therefore we are not concerned with the benign tumors out here. However, at the same time, we are much more concerned with those tumors which actually cause cancers, and therefore these are the tumors which are formed in the body, and there is a big challenge in treatment of these tumors. Basically, what are the common treatment process in cancer? So these are radiation therapy, chemotherapy, or you go in for surgical removal of the tumors. But what if we can target these tumors and it can actually destroy the cells inside them? So for that, these nanoparticles have been designed, and the nanoparticle penetration and stability are like within this microenvironment of tumor is very challenging, because the tumor microenvironment includes different characteristic properties like their vasculature, their makeup, then the fluidic pressure inside these tumors, and the extracellular matrix which makes up the tumors. So the density of the extracellular matrix, all these factors, that is the vasculature, the interstitial fluid pressure, and the extracellular matrix, all these actually contribute to the limited permeation, that is the permeability of the nanoparticles. So the nanoparticles cannot as such they move inside the tumors, but even if they move in, they have to fight out the vasculature, the fluid pressure, as well as the extracellular matrix. Now if you go into the next slide, you can see madam, the next slide. So you can see out here a particular ring shaped figure. So here, and actually we are putting the highlighting the barriers then a nanoparticle can overcome. That is in the inner ring. So you'll be finding, if you look in the inner ring where a human figure is there, so within that, in the on top of him is the nucleus and the cytosol. So you'll find that this the intracellular delivery. Then you come towards the right, you'll be finding that the navigating tumor microenvironments, then immune cells, then the epithelial barriers. So these are the important barriers which the nanoparticles have to overcome. And subsequently, uh, the role of nanoparticles, that is the application in which they can play, that is genome editing, if you look at the top. So then beyond genome editing, the treatment based upon biomarkers, then characterizing specific barriers to reach the target tissues. So after that, creating different kinds of cell therapies, then modulating the immune system, and determining the pharmacokinetics. So by I mean, these are all the application sites of the nanomaterials. However, the barriers which they have to face are also given in this particular diagram, and you can actually see out here what are the different kinds of barriers as well as the applications of the nanoparticles. Then we come almost near the last, so applications of nanoparticles. Ma'am, the next slide. So in applications of nanoparticles, the first thing that comes into mind is the cancer therapy. So nanoparticles have been developed for targeted therapeutics for treating patients who expresses specific biomarkers. So again, we have previously said about, I have previously said about how biomarkers actually developed, and based upon that biomarkers, how we can go in for treatment of the people with the particular disease. Now, novel nanomaterials, which are developed by incorporating cell membranes into the nanoparticles, they also improves the accumulation of the nanoparticles in the cancerous tissues. Okay, so this is again important because that means the nanoparticles have been developed which can actually move into the tissues and carry out their activities. Then nanoparticles can also be wrapped with macrophages or leukocyte membranes, and these can also recognize tumors and increase the specificity of the cell. So these type of nanoparticles which are actually like wrapped up with a macrophage, or I mean actually when we say about wrapping up with a macrophage, that means taken up by the macrophage or that is already being phagocytosed. Okay, so such type of nanoparticles, they actually show a rise in the activity, that is 2 to 3-fold rise in their drug activity. Again, nanoparticles are being designed to improve the percentage of chemotherapeutic dose, which makes I mean which helps in reaching the nanoparticles to the site of the solid tumors. So nanoparticles are again useful to like improve upon the chemotherapeutic dose. So this is very important, and this is most I mean mostly of concern because you know in chemotherapeutic, whenever we are given a chemotherapeutic drug, the patient, he or she actually is not that same self anymore, and there are a lot of changes which occurs in the person, and therefore if we can help in increasing the activity of the drugs and minimizing the side effects, then it can be a boon for the patient who is under treatment. So therefore nanoparticles can have to be designed or in such a way so that the percentage of chemotherapeutic dose is increased or improved in such a way that it helps in reaching the site of the solid tumor. The next important application is in immunotherapy. So nanoparticles have the potential to significantly improve delivery by protecting immunotherapeutics and enhancing the interaction with immune cells. So this is a major area of focus of nanoparticles, that is immunotherapy, where they actually improve the immunotherapeutics, and particularly in those diseases which are related to the immune system. Ma'am, next slide. Yeah. To improve immunotherapies where a T cell is targeted, and herein the efficacy and the side effects are minimized, and the nanoparticles actually have been formulated for the delivery of monoclonal antibodies. So monoclonal antibodies are very efficient. They are very much been used in the last 2 decades for treatment of different kinds of cancers, and these nanoparticle formulations, they also disrupt the immune checkpoints to the delivery of small interfering RNAs. So herein also it is important because the immune checkpoints actually prevents a particular drug or some important cellular events from being taking place. So they will also prevent the nanoparticle formulations from moving inside. However, that is what we do not want. So for that, we have to disrupt these immune system checkpoints. And therefore, there are certain small interfering RNAs which would actually stop the function of these checkpoints and thus lead to the formulation of the nanoparticles and delivering of the particular drug. Then in gene therapy, so inhalable nanoparticles have been designed and developed for delivery of mRNA in the treatment of cystic fibrosis, which is a genetic disease. And besides these three major topics, there is cancer therapy, immunotherapy, gene therapy. Nanoparticles are also being used in the ocular treatment of ocular diseases, kidney-related diseases, then in most of the other like metabolic diseases wherein diabetic related complications are also being resolved. So therefore, not only these three, but nanoparticles have made their inroads into different kinds of disease treatments, and they are being used all over the world, and they are being researched for making their applications into much diverse areas. So in this next slide, Madam, the next slide. Yeah, thank you. So in the next slide here, you can see that the nanoparticles, which have been engineered for their surface properties to increase the delivery. So herein, if you look into the green colored substance, that is a nanoparticle. So you can see how different kinds of substances have been put in. That is a self-peptides, the pegylation process is there, cell coating is there, then density and toughness is measured. Then again, we are putting in aptamers, then vitamins, then cell surface receptors can be done, then antibodies, then putting in the carbohydrate moieties, nosdextrin, etc. So these are different kinds of modifications which actually are carried out in a nanoparticles so that they can actually remain stable, perform the activity, and deliver the particular drug. So these are very important in terms of nanoparticle functioning and their role as a drug delivery agent. So we are moving to the next slide, madam, and the next slide. Yeah, thank you. So advantages. So we're almost at the end of the lecture, and these advantages actually are like overall all the things that we have taken into account till now, and why we should go for nanoparticle research or nanostructures in drug delivery, because nanostructures like liposomes help in the delivery of hydrophilic and hydrophobic drugs. So this is very important because both kind of drugs can be incorporated and taken in, both hydrophilic and hydrophobic drugs. So here in hydrophobic means those which actually hates water or fear water, and hydrophilic are those which love water. So these are particularly liposomes or the lipid mediated nanostructures which can help in the delivery. Then most of the nanoparticles have been found to be non-toxic or developed in such a way that they are non-toxic, but at the same time they are biodegradable and biocompatible. So biodegradability and biocompatibility are two of the most primary points for nanoparticles, and their non-toxicity is very important. So they should not have any side effects. Then the polymeric nanoparticles increases the solubility of the hydrophobic drugs. So again, hydrophobic drugs, that means those drugs which fear water, they will not be soluble in the system. So therefore, they have to be made soluble, they have to be made to work in that particular system. So for that, we have to design the nanoparticles in such a way so that it increases the solubility of the hydrophobic drugs. Again, not only increasing of the solubility, but also the cytotoxicity has to be minimized. Apart from that, the circulation of these nanoparticles must be enhanced. Now, These nanoparticles have to be again facilitated for their entry, and there has to be an improvement in their utilization. Now the thing is that when we are saying about the circulation time, because the more they circulate, the more they will be releasing the drug in an appropriate manner, and that will be actually taking up the entire process, and they will be actually helping in controlling of the disease factors, because herein slow release of the particular drug in question will come up, and slow release but determined dose, and that will be actually helping up the particular treatment process. Then the nanocapsules, which we have discussed before, these nanocapsules helps in the bioavailability of the nanomaterial, of the drugs, and it also protects the degradation of the drugs and help in the controlled release, as well as helps in masking of any unpleasant taste. So these nanocapsules, they not only protects the degradation of the drugs, but also helps in the controlled release of the drugs by masking and it also helps in masking of any unpleasant taste. So when we're taking it through the oral route, if the taste is not good, we are not going to take it, we will throw it out. However, these nanocapsules, they mask the drug in such a way that we don't actually get it, right? Then the mesomeric silica based ceramic nanostructures have cytotoxic properties against cancer cells when they are functionalized. That means when different kinds of functional groups are being added up. The calcium phosphate based ceramic nanostructures are again used in bone remodeling. So this is a very beautiful feature. And aluminium and titanium based ceramic nanostructures have been found to be used in drug delivery due to their high water dispersibility and stability. So they can disperse in the water very very easily. So they are being used almost in for any kind of drug delivery studies nowadays. Again, peptides are mostly used for their biodegradability and biocompatibility. So they actually form a well-defined nanostructure of different shapes and sizes. So peptides are, again, since they're biological in origin, so therefore they can be used. So that is actually an advantage. So remember, and the next slide, please. Yeah, thank you. The DNA-based nanostructures are, widely used in various modifications have been carried out to perform the delivery of different drugs and genes using these DNA-based nanostructures. They are basically nanotubes, nanoballs, nanofilms, then hydrogels, etc., which have been prepared. And these type of nanostructures, they provide flexibility of loading the drugs. Okay, so we can actually load different amount of drugs into these different kinds of modifications which have been made. And then we have spoken about graphene. So graphene is one of the most sought after materials because of its low cost, and then we can fabricate it very easily, and they have a very high drug loading ratio. So that means we can load in a very high amount of drug into this graphene, and with that actually we can improve upon its disease treatment possibilities. So again, graphene has a very good biocompatibility and low cytotoxicity, which is very important, and very unique physicochemical properties. Then the carbon nanotubes. So carbon nanotubes have been widely used as they have a large inner volume and an outer space, which can be modified to load proteins and genes. So carbon nanotubes are one of the most important features, and they have a large inner volume, so therefore you can incorporate a higher drug ratio again. Then the next is the inorganic nanoparticles, due to the magnetic, radioactive, and plasmonic properties, are applied on a variety of applications such as diagnostics, imaging, and photothermal therapies. So again, this application is completely different. It is not related to the system, putting out into the system, but it is related to the external parts, that is the diagnostics and imaging parts. However, in imaging, sometimes nowadays different kinds of injections with different kinds of markers are being given, so that the images of the particular organs are more visualized, they're more fluorescence, the picture quality becomes good, and determine the disease actually is more faster. Then functionalization of the nanoparticles allows the recognition of the specific site of action, avoiding high systemic concentration and reducing side effects. So the nanoparticles also helps in recognizing a particular action, type of action, and they also avoids systemic concentration that is going through a normal systemic way of circulation, and then reducing the different kinds of side effects. So therefore functionalization is very necessary. Functionalization means putting out different types of functional groups on top of the nanoparticles. That is, we are dressing up the nanoparticles. Okay. Then madam, the next slide, disadvantages. Yeah, thank you. So nanoparticles, although they have various advantages, yet they also have a lot of disadvantages, and that is why it is a hot topic of research which is going on since the last 2 decades. Had there been no disadvantages till now, till date, we could have seen a lot of nanoparticle based medicines in the market, although till date there are only a few of them and research is still going on. So what are these disadvantages? So disadvantages are like, in case of the liposomes, the membranes are very thin, fragile, and therefore they are not stable. So stability comes in here. Okay. Then they have low encapsulation efficiency and poor storage stability. So we cannot keep them for a long term. We cannot store them for a long term. So that is again a disadvantage, high disadvantage, because cost wise, if you see, if we do not use them within a short period of time, they will get expired. And even in the body, once you have put them, if they're not that stable, then the drug in question will not be targeted, okay, or will not be delivered into the particular target organ, target organ or the target disease factor. So due to low permeability of the nanocapsules, there is a problem in encapsulation and release. So again, in the case of liposomes and in the case of nanocapsules, there is also a problem in the encapsulation and release factor. Then ceramic nanostructures, which are which are those prepared using non-biodegradable ceramic nanoparticles such as hydroxyapatite, alumina, and titania. These can accumulate in the body and cause severe side effects. So we have to be very much concerned about the type of material that we actually use in preparing the nanostructures, and we should see that we do not use those type of materials which actually pose a problem in us. Then uh, in case of metallic nanoparticles, the silver nanoparticles are not much preferred due to the reported toxicity against eukaryotic cells. So you can see that in majority of the cases, we are using the gold nanoparticles and not silver nanoparticles, as they have been found to to be toxic against the eukaryotic cells. Now, in case of DNA-based nanostructures, various limitations exist because DNA-based work are always very costly, and therefore they also have low bioavailability. So this is another major area of focus. There is the bioavailability of the DNA-based nanostructures. And the scaling up cost and safety concerns are few of the disadvantages of various nanostructures. So we just cannot think about synthesizing them, but they scale up. Okay, there is manufacturing time in the large amounts. So that actually involves a lot of cost factor. So that is a major area of disadvantage, and people are dissuaded, scientists are dissuaded. So therefore, there has to be certain times, government backup subsidies, which can actually help in, taking up this kind of entrepreneurial ventures, so that this disadvantage can be made into an advantage. Ma'am, the next slide, please. Okay, so the surface properties of nanoparticles can induce inflammation and mediate allergic reaction. So you see, we should not be utilizing those materials in the surface coating, which can actually pose a problem in us later on. Because if the surface properties can induce inflammation and induce allergic reaction, so again, our immune response will be promoted. So which in turn will have a, in the long run, it will cause inflammation. So this inflammation will again have problems towards the our own cells, and they will in turn make them cancerous in the long run. So this should not be warranted. So therefore, when we make nanoparticles, we should see that the surface are usually made with such kind of materials which will not have any such problems in inflammation or mediating allergic reaction. Again, inorganic nanoparticles have various advantages, but they are limited in the clinical applications due to this low solubility. So again, inorganic nanoparticles, previously what we have spoken about, like gold nanoparticles, then iron oxide nanoparticles. So they have low solubility and toxicity, mostly in case where the formulations uses heavy metals. So we have to be again look into this type of restrictions. That is, heavy metals should not be used, and if heavy metals are being used, we have to study the toxicity before being incorporating them into a human system, or before going into the trial runs. We should sorry, before putting them up in the market, we should always go for trial runs and see the toxicity that they incorporate, and to remove that, what we should do. Next slide. Yeah, so we are almost concluding the talk. So the nanostructures, due to the wide range of uniqueness and valuable properties, are used in the drug delivery process, which we have discussed in over this last 2 hours. And the nanoparticles can be optimized to improve the drug bioavailability. So we have to carry out this process, and we can actually do that ourselves. So in the process of synthesizing the nanoparticles. So herein, how we can do that is by increasing their absorption through enhanced solubility, or by facilitating their passage through the biological membrane. So these are the different ways. Then again, various advantages such as the increased solubility of poorly soluble drugs, then making them making them more like user friendly, like having less side effect, and then increasing the efficacy, can make the nanoparticle based drug delivery a widely acceptable choice. The nanostructures are very structurally diverse, and therefore we have a lot of wide choice of materials which we can make. So we can select our own starting materials from which we can actually prepare these nanomaterials, and based upon their efficiency, we can actually help them in by loading the particular drug, or even by going for functionalization of these nanoparticles. Again, although a lot of nano-based medicines have been developed, it is far below the projection, as there is a translation gap between animal and human studies. So a lot of, actually, before going into the market or putting the drug in the market, a lot of human and animal studies have to be carried out. Nowadays, of course, in India, because of stringent rules, we cannot go in for a lot of animal studies. So that is a major problem. However, again, we should be considerate enough towards the animals. But then, for the advancement of human science, I believe that we always need such kind of model organisms where we can study the drug efficiency. And also, before putting into the market, a clinical trial must be there. And clinical trials of the various stages, 1, 2, 3, 4 stages, have to be carried out. And during this time, we also have to study about the side effects in humans. If these drugs or these nanoparticles actually pose a lot of side effects, then we have to take them back and do research in them so as to see if they can be made free of those side effects. Now, again, when you're speaking about animals, it is not that animals have to be killed. No. Similarly, just like we are treating humans, animals have also to be treated in that way. So only the science will advance, otherwise it will not. Then, biodegradability and toxicity of polymeric nanoparticles and a formulation still require further absorption to have an accumulation of polymeric carriers after repeated dosing. So this thing is very important, because when we apply medicines or apply drugs, we give them at specific doses. So it should not be that they will be accumulating, right? So if they accumulate enough, they will only pose a problem for us. So this accumulation has to be prevented. So therefore, they have to be cleared after they have performed their functions. Madam, next slide. The biocompatible polymeric nanoparticles have been thus synthesized and developed, and we can see the various processes by which they have been synthesized and being developed. And the novel systems which are biodegradable and can be synthesized from various resources like artificial and natural resources are the need of the hour. So we have to find out, do research in actually the novel systems, try to find out much more unique systems which will be better for us, better for these studies. Then in case of gene therapy, nanoparticles have the potential to improve genome editing by exerting more precise control and reducing safety concerns. So nowadays we have all heard about the CRISPR-Cas9 genome editing tool and how actually nanoparticle research have also been clubbed with CRISPR-Cas gene editing tools to perform better gene editing functions and treat various genetic diseases. Then the process used for nanoparticle uptake is determined by numerous factors, including characteristics of the cell membrane as well as the properties of the nanoparticle, which also influence the subsequent endocytic process. So this is what we have discussed, like the way the nanoparticles are being made actually plays an important role in their in their uptake, and using again various factors which actually will have important role in the stability of the nanoparticles, and finally the delivery of the required drug into the appropriate target organ. So this is what I have to present today in this meeting. In the next slide, you can see actually some of the references that I had actually went on. Madam, this is the DOIs have been given, and there are some of the very good papers. Actually, I had checked on some more, but these are the ones which I had extensively followed. And you all can also have look into these papers, and there is one Nature review paper, the number 2 1, so you can look into that. So that is very beautiful. It's just 2021 publication, so very recent publication. And madam, the next slide please. But I thank you all for your patience, and I have taken exactly 2 hours as has been suggested to me, and I thank you all for this, for your patience in hearing me, hearing my lecture. So if there are any questions, I think I'd like to answer.
Thank you very much, sir. Thank you very much for the informative session focusing on polymer-based nanostructures, polymeric dendrimers, ceramic nanostructures, biocompatible inorganic compounds, peptide-based nanostructure, metallic nanostructure, nucleic acid-based nanostructures, carbon-based nanostructure, covering all the nanostructures, methods of preparation of polymeric nanoparticles, uptake of nanoparticles, delivery and biodistribution of nanoparticles, and the recent trends of nanoparticle circulations. And the genome editing, one of the recent technology and applications in the biomedical field like immunotherapy, gene therapy, then along with the advantages of nanostructures and disadvantages. Thank you very much, sir. Now the session is open for interaction. If any participant says anything to interact with sir, to ask sir, the session is open. Please raise your hand.
Madam, you can also share my email ID, like if there are any questions which they would like to ask me over emails and everything, so they can actually go for that also to the participants.
Sumit has raised a
Yeah, sir.
hand.
I'm asking in India, where is going on this type of therapy by nanoparticle in India? On in any aims or etc.
Okay, so actually, nanoparticle research have been carried out in most of the IITs, in IIScs also you can see out. But actually, in pertaining to human clinical trials, most of the studies are actually being carried out abroad. So which,
Abroad.
yeah, most of them.
No,
So because
not in India. Not in
no, no,
India.
I mean,
This time.
see, this time it is, I cannot say it appropriately and to the particular institute which is carrying it out, because the papers that I had went on or focused on, I could not find any relation to their reporting about, doing out research or carrying out actually research in the level of clinical trials of drugs pertaining to nanoparticles. So in India.
But sir, in India, it will be successful this type of
Yes,
therapy
yes,
because
yes.
no,
It will be successful.
because if I talk about many people's living in a general way, not earning more, and if I talk that manual particle cost is very high in a lakh or 15,000 in a small
Yes, yes,
amount.
yes. That is true.
So according to you, it will be successful this type of therapy in India.
See, the first thing is the raw material purchase and its product, the final product in the market is
Yes.
totally different, because the raw material that that is being manufactured is very high in cost. Some of the raw materials, not all, I'd say.
Yeah.
If you take
Yeah.
graphene and all, so these are quite cheaper. But if we go for gold nanoparticles, so that is of course quite high. Okay, so
Yeah.
again, the products that are made out from these particular raw materials, they will also vary in the price. So therefore there is a different, this final from the initial startup in the laboratory to the final product in the market. So subsequently, if at all in India, in a nation like us, it has to be a success, we have to look into the cost view. The cost view of the final product and the treatment process using that. Because like if it costs you 10 lakhs for treatment using a nanoparticle,
Yeah.
only those people who have enough money can be actually cured, but not the stable general population who do not have that much of uh, worth to get the treatment. So this is a limitation. Yes, of course it will always be there, unless and until, I believe, the government comes into play, and they can actually put in some stringent rules. And yes, one more thing is that many of the nanoparticle raw materials that we actually take in for our research are being synthesized abroad. We are not having that much of companies which are doing it in India, because the raw materials are available outside. We are only doing in some, for some cases, but not for all. So therefore, when we are bringing it, we are importing it, the cost automatically shoots up. So therefore, at the end of the day, our products become costlier. So that is a big problem. Yes, that is true.
Yes, sir. More time will spend to come in India. More
Yes,
more year.
yeah.
Okay, because the instrument also is lack in India
Yes,
to check the stability to optimum condition of checking the optimum condition of nanoparticle.
yes, true. All the
Okay, thank you,
instruments,
sir.
related to nanoparticle research are also very costly. That is very true.
Costly and very number of instrument is very less this time.
Very less. And apart from that, you also need expert people in handling those equipments.
Yes, sir. Yes, sir. Okay, thank you, sir.
Yeah, thank you, Dr. Sumit. Yeah, thank you.
Oh,
I think this is the global scenario of nanotechnology, the market.
Yes, ma'am.
Because
That is true.
whatever technology we are taking, those are related with market. So global
Yes.
nanotechnology, maybe after 10 years, it will be heading in this direction in
Maybe,
biomedical.
yes, ma'am. That is true.
So if there is no questions, I think there is not more questions. So thank you very much, sir. Thank you very much for giving
Okay.
your valuable time and sharing this informative session with all of us. We are greatly benefited from this.
Thank
And
you, ma'am.
on behalf of CIPET CSTS Guwahati, thank you for giving this expert session to us. Thank you, sir.
Thank you, ma'am. Thank you for the opportunity. Thank you.
Yes, sir, you can leave now, sir.
Okay,
And
okay.
another one is that uh, the participants are asking your email address.
Yes, ma'am. It is dkranjan at
May I
gmail.com.
share your
Yes,
communication
ma'am, please.
information, contact details
Yes, ma'am.
with the
Yes, ma'am.
participants?
Yes, ma'am.
Yes, sir. Okay, sir. I will share.
Okay. Thank you. Thank you.
So, dear participants, uh, Thank you very much. And our session tomorrow will start at 9:30 a.m. And tomorrow the first session will be by Dr. Yasbir Singh sir from IIT Ropar. He will talk on polymeric hydrogels and gels for drug delivery and antibacterial applications. So I request all of you to join by 9:25 a.m. Thank you so much. You can leave the meeting. Mail. About 2 minutes.